Welcome everyone, and now let's start the next topic of the analytical chemistry, that is the atomic emission spectroscopy, which is abbreviated as AES. The word spectroscopy, now you are quite familiar with it, that the study of the interaction of electromagnetic radiations with matter, that is called as spectroscopy. The two new words in this spectroscopy are atomic emission. We will discuss these two words in detail today, and we have again divided this lecture into the different parts which are in front of you. So, without wasting time, let's move into the topic. First of all, we will discuss the introduction of atomic emission spectroscopy. The first word is atomic, second is emission. It means this spectroscopy is related to the atoms, and there will be certain radiations that will be emitted from the atoms. When we talk about an atom, we know that an atom has a central portion which is called as nucleus, and then there is an extranuclear part of the atom which consists of different shells, orbits, or energy levels. So these are different energy levels that an atom has. Keep in mind that every atom, even it is an atom of hydrogen, every atom has infinite number of shells or orbits or energy levels. In this particular lecture, I will be using the word energy level frequently. I will not use the word shells or orbits, although they also mean the same. Just because of the reason that this whole topic is based on the energies of the shells. So I will use the word energy level. The number of energy levels in any atom, that is infinite. Now, when we say valence shell of an atom, that never means the last shell. That means the last occupied shell. Mean the shell, highest shell having some number of electrons in it. That is called as valence shell. Otherwise, the number of shells in any atom, they are infinite. So there will be no concept of valence or lost or the last shell. No. Instead of showing these energy levels in circular form, I have shown these energy levels in this forms, in the form of lines. This one is energy axis that will show that here is the lower energy, and as we move up, the energy will be increased. And these are energy levels of two different atoms. Suppose we have an A atom and there is another atom which is B atom. Now you can see that I have shown 5 energy levels of each of these two atoms. But the first energy level of A atom, and if you compare it with the first energy level of B atom, it is quite evident that both these energy levels, they are having different energies. Similarly for the second, third, fourth and fifth. So the first point that you must keep in your mind, that atoms do consist of energy levels, but the energy levels of the shells of the one atom, they are quite different from the energies of the energy level of all other atoms. Actually, why these energies of these two energy levels are different? Because the energies of the shells or orbits or energy level, they depend upon the electron-proton ratio of an atom. When we deal different atoms, every atom of different element that has different electron-proton ratio. So its energy levels, they will have its unique energy distribution. A atom, B atom, and if there were any C atom, even the first energy level of the C atom would be quite different with respect to energy as compared to the first energy level of A and B atom. So energy levels have different energies in case of different atoms of the elements, different elements. Similarly, you can see that the energy difference or energy gap between these energy levels, that is also different in both cases. This delta E is quite different from this delta E. So this one is the second point. There are only 2 points. If you keep these 2 points in your mind, the whole concept will be clear. Atoms consist of energy levels, and the energy of those energy levels in different atoms, that is different. Every atom has its own unique pattern of the energies of its shells. And because the energies of the individual energy levels are different, so the gap between the energy levels, that is also different. And in case of different elements, different atoms, there will be different gaps of the energies between the shells and energy levels. That is about the introduction of the atomic emission spectroscopy. Now, what is the principle? Suppose there is an electron here in the first energy level, and you provide energy to this A atom. That energy may be provided in the form of heat or in the form of radiations. When you provide energy to this A atom, that energy will be given to, or that energy will be moved to this electron. And this electron will move from, suppose it is moving from first energy level to the second energy level by absorbing certain amount of the energy. When an electron moves from low energy level to high energy level, we say that the electron is excited. So this is excitation of the electron. Similarly, there is an electron here in this atom, and when you will give energy to this atom, the electron will move from low energy to the high energy. The electron is excited. What do you think? Will both these electrons absorb same amount of energy during their excitation? I don't think so. They will not absorb same amount of energy. Why? Because the energy gap that they are covering, that was different in both atoms. So these two electrons will absorb different energy, different photons of the specified energy when they will be excited. But you know, every electron, when it moves from low energy level to higher energy level, it becomes unstable. And in higher energy level, it can spend maximum of 10 raised to power minus 14 seconds. In some books, it is written that electron can spend maximum of 10 raised to power minus 8 seconds in the higher energy level. So, keeping both in mind, 10 raised to power minus 8 or 10 raised to power minus 14 seconds, it is quite evident that whenever an electron will move to higher energy level, it will spend very short duration of time there. And this electron will again come back from higher energy level to lower energy level. And when it will come back from higher energy level to lower energy level, it will emit same amount of energy as it absorbed when it was excited. Similarly, this electron will also move from higher energy to lower energy, and it will also emit same amount of energy as this electron absorbed during the excitation. When both these electrons were excited, they absorbed photons of different energy. And when they will be de-excited, according to law of conservation of energy, they will release photons of the same energy. So when energy absorbed was different, so energy emitted will also be different. This is the base, this is the principle of the atomic emission spectroscopy, that when the electrons are excited in the different atoms, they absorb different amount of energy, and when the electrons are de-excited, then they release different amount of energy. Now the energy absorbed and released by atom of one element, that will be different from the energy absorbed and released by atoms of all other elements. So in this case, these emitted photons will be characteristic feature of every atom that will be different from all other atoms. And when these emitted photons will be observed, studied using emission spectrometer, the emission spectrometer will make emission spectrum with the help of these emitted photons. So because these emitted photons are having different energy, different frequency, different wavelength, different wave number, all these quantities will be different. So the atomic emission spectrum or emission spectrum in both these cases, in case of A atom and in case of B atom, that will be different because the emitted photons were different. This is the principle of the atomic emission spectroscopy. So now it is quite clear that why it is called as atomic emission, because the electrons during their de-excitation they emit photons. So that's why it is called as the atomic emission spectroscopy. Now let's move towards the next part, that is the samples that can be studied and sources of excitation. So first of all, if we talk about the samples, what kind of samples we can study in the atomic emission spectroscopy? We can study a sample in gaseous form, in spray solution form. If there is solution, that can be used in the form of spray with the help of an atomizer, or even the solid sample can also be analyzed, but in order to analyze this solid sample you will have to change it into slurry or paste. So gaseous atoms in the form of liquids, spray solutions or solutions in the form of small mist, and the solid, but solid sample in pure form cannot be analyzed. You will have to make its paste by using some solvent. So these are the samples that can be analyzed by the atomic emission spectroscopy. Now what are the excitation sources? Excitation sources mean what can be the sources that can cause excitation of the electrons. So you can use flame, as you are familiar that you use flame in the flame test. You can use an arc or spark can be used, or even plasma. Plasma, you know, that is a very high energy state. So the energy of the plasma state can be used for the excitation of the electrons in your sample. These three are the ancient sources. Mean they have been used since long. But nowadays in the modern atomic emission spectroscopy, the sample is excited with the help of the plasma, and that plasma is generated using the argon. So these are the samples and excitation sources. In which form samples can be used and what high energy sources can be used in order to excite the electrons in the atoms. Now let's move towards the next part. That is the technique. Now what technique is used? I am just making a schematic diagram in front of you people to clear this concept that how atomic emission spectroscopy works or how atomic emission spectrometer works. Suppose you will just have to suppose it that it is flame and if your sample is in gaseous form or it is in form of spray solution, you can use atomizer. What this atomizer will do? it will produce fine mist of your sample, and that mist will be thrown on the flame. The flame is the source of the excitation. But if your sample is slurry or paste of a solid substance, then we can use, as you know, that we can use a platinum wire having a loop at its end, and here that slurry or paste is introduced, and this slurry or paste is placed on the, or this loop is placed on the flame, and flame causes the excitation. So this is a way to introduce your sample to the flame. Even you can use the discharge tube, that we have studied that how the discharge tube works. But I am here focusing on the flame atomic emission spectroscopy. Now when the electrons will be excited, as I have already explained it, and when those electrons will be de-excited, they will emit certain photons of the specific wavelength, specific energy. Then we use a monochromator, or I am here showing a simple slit. This slit will be the wavelength selector. It will select specific wavelengths and it will let them pass. Now when these photons will move forward, here is the prism. You know, the prism causes the diffraction of the radiations, and that diffraction depends upon the wavelength of the photons. After passing through the prism, the photons or the light or the electromagnetic radiations, they are dispersed. And when they are dispersed, there is a plate on which a visual display is produced, and that visual display is called as the atomic emission spectrum. Now this atomic emission spectrum will be having spectral lines, and these spectral lines will be bright while the background will be dark. But keep in mind one thing I have already told you, that during de-excitation, every atom of the different element emits different photons having different energy frequency. So the position of the spectral lines will also be different in case of atoms of the different elements. So atomic emission spectrum of an element is its characteristic feature, and the atomic emission spectrum of one element is very different from atomic emission spectrum of other atoms of the different elements. So by looking at the atomic emission spectrum, by looking at the position of the spectral lines, by looking at the colors which are present in the atomic emission spectrum and the scheme of the colors, we can easily identify the atom which we are analyzing, which is our analyte. So this is a technique and how the atomic emission spectrum is produced. Atomic emission spectrum has bright spectral lines because these are the energies which are emitted. These are the photons which are emitted. So the emitted photons appear in the form of the bright spectral lines, while those which are absorbed, they will have the dark background. So the background will be dark and the lines will be bright. This is the technique which is used for the detection of the elements, for the detection of the atoms of the different elements, because atomic emission spectrum of every element, every atom, that will be different from all other atoms depending upon the photons that were emitted during the de-excitation of the electrons. After technique, now we are moving towards the next part, that is atomic emission spectroscopy versus flame test. You know that flame test is also used as a preliminary test for the detection of the metals. Then, if flame test can help us to detect the metals, why we use the atomic emission spectroscopy? The flame test has certain disadvantages. Suppose that this is flame, and you know that you take a platinum wire and at the end of the loop you place your sample sorry, in the form of slurry or paste, and when you heat it into this flame, the color of the flame is noted, and in case of different metals the color of the flame is different, so we can detect the metal. Then this flame test is very easy. Just heat the loop and note the color. We can detect the metals. Why do we use then atomic emission spectroscopy, and why do we make the atomic emission spectrum to detect the metals? This technique is especially for metals. The answer is that there are two drawbacks of the flame test. Number one, colors get mixed. When you heat an atom, when you heat your sample has collection of atoms in it. And when you will heat those collection of atoms, in case of different atoms, the electronic excitation will be different. Suppose in one atom, the electron excites from first shell to the fifth. In the other, the electron excites from first shell to the fourth. So different electrons will absorb different energy and their excitation will be different. When they will be de-excited, the photons of different frequencies, different energies will be produced. There will be mixture of the photons, there will be mixture of the colors, and sometimes it becomes very difficult for us to distinguish or to notice that color, that which color is going to be produced. Sometimes it's become violet, then a slight shade of the blue is also introduced in it. So sometimes the colors, to detect the colors, that becomes very difficult for us in case of flame test, and that is due to the range of the frequencies, range of the energies that is released during the de-excitation of the electrons. And even sometimes two different elements, they show same color, just like lithium and strontium, I think so, I do not remember it, but both I think so show reddish color in the flame test. So when both will show reddish color in the flame test, how we can distinguish between them? It becomes very difficult. But in case of atomic emission spectrum, the position of lines, the pattern of the lines will be quite different in both the elements, so we can easily identify them. The second disadvantage is flame energy. Sometimes the flame is not having enough energy to excite the electrons in atoms. The energy of the flame is not sufficient for the proper excitations that we require for our analysis. So this becomes the second disadvantage of the flame test. But in case of the atomic emission spectroscopy, I discussed with you the flame atomic emission spectroscopy because that was quite a simple case. But nowadays plasma is used, and plasma gives so much energy that are required, and electronic excitations, they can easily take place, and we can observe what we want to observe actually. So this was atomic emission spectroscopy versus flame test. Now what are the applications of the atomic emission spectroscopy? The atomic emission spectroscopy is basically qualitative and quantitative. How? When I say that it is qualitative, it means by looking at the atomic emission spectrums and by looking at the patterns of the spectral lines, I can identify the elements, especially metals. How it is quantitative? By looking at the intensity of the photons which is being released during the de-excitation of the electron, I can judge how many atoms of this metal there will be in the sample. So by looking at the quality of light, qualitative analysis, by looking at the intensity of light, I can judge the number of atoms of the samples, of the sample, that how many atoms of a specific metal can be in the given sample. Even some non-metals like sulfur, phosphorus and iodine, especially these three non-metals, they can also be analyzed by using the atomic emission spectroscopy. And the concentration of iodine in the seawater, that is especially determined by using the atomic emission spectroscopy. I can also study the, or we can also study the mixers. and mixtures of 6 to 30 elements. That can be easily studied by using the atomic emission spectroscopy. Let me tell you how the atomic emission spectrum of the mixers will be studied. When there will be a mixer, the atomic emission spectrum will be very complex. But we will compare the atomic emission spectrum of the mixer with the individual atomic emission spectrums of the different elements. And then by looking at the arrangement of the spectral lines, we can easily identify that what elements or what different types of elements they are present in a mixer. So we can also judge the 6 to 30 elements. Especially when we are talking about the alkali metals, and we want to study them in the pharmaceutical studies, especially because being a student of pharmacy, I also know that they are quite, atomic emission spectroscopy is quite important for the detection of alkali metals during the processing of the pharmaceutical products. So these are some applications of the atomic emission spectroscopy. Beside these applications, there are so many others. You will read those applications from your respective books. And what are the merits and demerits of the atomic emission spectroscopy? Merits versus demerits. What can be merits? It is rapid, it is sensitive, mean micrograms of the samples can be analyzed even. It is selective and multi-elemental analysis or multi-element analysis. From mixer, as I have already told you, that mixers can also be analyzed. And what are the demerits? Especially the demerit is high cost of the apparatus, then difficult maintenance, then sometimes the interference of source of excitation. If you are applying plasma as source of excitation, then even the spectrum of that plasma or the excitation or de-excitation of the electrons in that plasma, which is used as a source of energy, that interferes with the atomic emission spectrum. So these are some drawbacks of the atomic emission spectroscopy. And keep in mind again that this lecture is for the beginners. This lecture is for those students that are studying the atomic emission spectroscopy at the 11th or 12th grade level. Otherwise, this technique is very wide, having huge concepts, huge applications. The time that I have taken in this lecture, even this time can be used just to explain one application of the atomic emission spectroscopy. I hope so that this lecture will help the beginners to understand the basics of the atomic emission spectroscopy. Next time we will come up with a new topic. Till then, Allah Hafiz.